understand that I really did not want to spend 2 days in a row going through material with you. But here's why I'm doing it. Because I think it's better to give you all the information now, and then you have until Friday for our test, right? Versus we're doing a lab tomorrow, and then it would be Thursday before I actually got that last bit of material to you. Does that make sense? So I figured it's better. And I realize the thought of listening to me again probably makes you want to throw up. I understand. Sometimes me too. Okay? But I think this is pretty solid. I think we'll be able to zip through this. There's very little, at least, I don't think there should be much that you all don't know about this. You with me? A lot of this is very, very much review. I want to hit really quickly the last little piece. Oh, I've got your study guides. You want those? Please. It is up on Schoology. The new study guide piece is up there. And it really didn't end up adding a ton more. So that's a good thing. Just give it to me afterwards, can you? It is on Schoology, yep. Yep. It is on Schoology. Thank you. Please. Extras, eh? All right. Okay. As we were finishing up yesterday, talking about how proteins were processed and secreted, and we were talking about the genes that help control that, we did not mention the fact that sometimes genes can be turned on and off. Because remember, only about a third DNA in any given cell is being used, right, to actually make proteins with. But it can be controlled when those get turned on and off. Sometimes they only come on initially, like maybe as you're growing as a kid, or maybe it doesn't come up until you hit puberty, or maybe not till later in life, or maybe it just comes on in various seasons. It just depends. But those genes can be turned on and off. That was kind of the key that I wanted to make sure. A great example of that is the mammary glands, right? The woman has the ability to produce milk at any time, but the actual producing of that milk doesn't occur until after the baby is born. Are you with me? It's very specifically timed. And there's lots of steps, so we'll go through that. They showed you in particular this, and I mentioned this yesterday, about this hormone, this prolactin, that the hormone comes and attaches to the receptor that's there in those mammary gland cells, and that produces this cascade that allows the cell to make the proteins to make the milk. Are you with me? But that's controlled by the hormone. The hormone turns on that gene. Maybe you were wondering, since we've spent a whole bunch of time talking about proteins, right? How we make proteins, all the process involved. Well, Mr. Hess, what about carbohydrates and lipids and nucleic acids? And we are 4 types of molecules we learned about. You spent all this time talking about proteins. What about the other 3? The good news is your body can make the other pieces, right? Glycogens and fats and steroids and phospholipids and all those other things. And it uses enzymes to do that. Okay? Where it'll break down the things that you've eaten and then rebuild things that you need. Does that make sense? So in order to process the other pieces, again, other than just proteins, to synthesize the other things, we use enzymes. That's the key piece there. And we're breaking down what we bring in through enzymes and building things back up via enzymes. Sounds like a good test question, right? Right, enzymes are what do that. Yep. They're synthesizing all the other things other than proteins. Okay, DNA replication. Let's talk about that. Are you comfortable? In order for a cell to grow, it literally splits itself into 2, right? And these 2 cells both need a copy of the instructions to make all the proteins it needs to survive, right? And so therefore we have to go into this process that we call DNA replication. So that the daughter cell, meaning the cell that it comes from the parent cell, right, actually has the controls. And we want this to be an exact copy, right? Because if I don't make it exact, there can be problems, right? And so what happens is a pretty straightforward process. 4 easy steps: unwinding, unzipping, building the new DNA strands, and then repackaging. So first, the DNA actually unwinds from the histones. Remember, the histones are like the little round guys that they loop around 1.5 times, so that kind of coils it up. So they unwind from there, and then this enzyme, DNA helicase, comes and it literally unzips. That's the word, because it's just like a zipper. Right? I should have had my sweater on today that had the zipper. But you know, you're unzipping it so that now instead of having 1, we have 2. Right? And that allows access to those bases, those nitrogenous bases, right? The CTAG. Then another enzyme, DNA polymerase, comes in and starts copying that. Not really copying, I shouldn't say that, because it's not really copying it. It's pairing up extra bases with the bases that are now exposed, right? So if I have an A on the DNA strand, it's going to pair a T with it, right? If I have a C, it's going to pair a G. Are you with me? And we just keep rolling down. And the same thing's happening on the other side of the zipper, right? So I'm literally making 2 new molecules of DNA that each of them have 1 strand of the old one and each have 1 strand that's new. In the nucleus. Yep, DNA never leaves the nucleus until the cell itself is totally replicating. Then the nuclear envelope breaks down. We'll talk about that later today. Does that make sense? And they have a big fancy name for that, that each DNA has 1 strand of old DNA and 1 strand of new. They call that semi-conservative replication. In other words, we're still conserving what we had, but only partially, right? Each of the new pieces get 1 strand of old DNA and 1 strand of new. Does that make sense? And then we take and we repackage it back up. It takes about 6 to 8 hours for the whole strand, that whole 2-inch strand of DNA to be replicated, which is pretty quick. I mean, on a molecular level, that's pretty fast. And then the DNA starts winding itself back up around the histones and folding back into that complex pattern we talked about yesterday. So here's a picture that you can see that. Here's the DNA being unzipped by the DNA helicase. And then you've got 2 strands. And here's the DNA polymerase that are coming along and taking these extra nucleotides, these spare pieces, if you will, and using them to make the other side of the DNA strand. So each daughter's DNA has 1 old strand and 1 new strand. Does that make sense? Are you comfortable that there are errors that are made in this process? What do we call those errors? Mutations, right? Some mutations are good. Some mutations are bad. Normally, they're more on the bad end of things, okay? But theoretically, you could do something that would actually make things easier for it to work. Now, the body is amazing. Actually, the Nobel Prize 2 years ago was won, maybe even last year, was won in chemistry for finding out about these DNA polymerases that literally go through and check for errors and fix them if there's something messed up, which is pretty impressive. And so the result is only 1 error per billion bases replicated, which again, if you only missed 1 question out of a billion, I mean, that's pretty solid, right? That's pretty solid, okay? And again, so sometimes the DNA changes, the mutations happen because of this replication error, but you can also get them to be affected by environmental factors. Like if you have a certain virus, it can cause changes in the DNA. If you have radiation that you get, like a nuclear bomb went off, or you had too many X-rays, you would have to have like Madame Curie with her X-rays, right? Just going to the doctor, you're going to get a bunch of X-rays. But the point is that environmental things can also cause problems. Any questions there? That's DNA replication. So mutations can be positive or negative. Mutation just means something has changed in the DNA. It can be because of an error in the actual replication process. It can be because of these environmental factors. All right, so let's talk about the cell cycle. Oh, this is going back to biology, right? You remember this? That you've got really two different phases. You've got the interphase, and then you've got the mitotic phase, where things are actually splitting. The cells are actually reproducing themselves. I will mention, because I put it in the study guide, that you have meiosis and mitosis. These are the only two ways that our bodies will make new cells. Most all of our cells do which one? Meiosis or mitosis? Mitosis. What cells in our bodies do meiosis? The egg or sperm. And the point of that is because we don't want a complete cell. We only want half a cell. We only want half of the chromosomes in these eggs and sperm. So that when one comes together with an egg comes together with a sperm, you actually have one whole person instead of like some mutant large person. I don't know. It wouldn't work. Does that make sense? But everything else uses mitosis. That's what we're going to focus on here. We'll save the meiosis conversation for the reproductive system down the road. So in interphase, you've got this G1 and G2 in the S phase. I'm not super concerned about this. Interphase is just the cell doing its normal work, right? In that synthesis phase, right here, the blue, that's where the DNA is replicated. So it's preparing for mitosis, but it's still going about its normal work, right? The cell is still doing normally what it does. It's only when we get to the mitotic phase, this mitosis area up top here, that's where the cell is actually stopping what it's normally doing so it can split into two cells and then continue again. So in mitosis, and why do we do mitosis? What's the functions of it? Well, because you're going from literally one cell, right? The sperm and the egg together, that's what you started as, to an individual that has roughly 50 trillion cells in their body. All that came from those one egg and one sperm, that one cell together. So we've got to make you got to develop, right? You're growing the tissues and the organs after birth. You're replacing cells that die. Are you comfortable that you replace every cell in your body every couple of years? You are not the same person you were 3 years ago. Like literally, you are not the same person, right? Everything replaces. Everything changes out. And also there's damages, right? You hurt yourself, your body's got to fix it. Sir? Sure. Yes. What I'm saying is that you have no cells or very few cells in your body that were there when you were born. There's a couple of exceptions to that rule, but that's what I'm meaning. That every cell is replaced. Yeah. If you just were to look at the cell's DNA of each of them, look at the code for you and the code for me, they'd be almost identical. Which makes sense because we're both humans, right? We both have 2 arms and 2 eyes and 2 legs. And no, the G0 phase is basically where the cell has reached a dead end. That the body has decided, hey, you don't need to reproduce anymore. You're just going to die. We'll let the other cells take care of it. You're being put out to pasture. That's what that G0 is talking about. Okay, so, and again, you've heard this 1,000 times, right? Prophase, metaphase, anaphase, telophase, right? So the big picture is what I want to make sure you're comfortable with these guys. Okay? So in prophase, I'm going to use the pictures if you don't mind, because I think it's easier to see. Where are we at? We're here on page 129 if you want to look at it in your textbook. Prophase, we're taking a cell that is a normal cell and we're preparing it to divide. So Where are the chromosomes normally located in the cell? In the nucleus. But we need to get them out of the nucleus in order to actually divide, right? The nucleus is in the way otherwise, right? So therefore the nuclear envelope breaks down. The actual outside of the nucleus breaks down, comes apart. So the chromosomes can unwind and spread their wings, right? Get bigger. Sir? Yeah, it'll just be like the pieces will hang out in the cell and wait for it to divide, then it'll come back together again. It's a good question. And I shouldn't say, I said that the chromosomes will expand. It's actually the other way around, and I apologize for that. Thinking about, remember, the chromosomes are these guys, right? The little U's. And in that chromosome is made up of the chromatid, right? The one little strand. So that is normally fairly relaxed, right? Because we got to get enzymes in there to copy the recipes for the proteins, right? But in order to reproduce, it condenses into a more smaller form. Does that make sense? So that's really where we see these chromosome-looking things come out when the cell is reproducing itself. Does that make sense? And again, the DNA is already replicated, so you see it's already got these two daughter chromosomes together, right? It's actually two chromosomes that are attached together into one piece. So that's prophase. The chromosomes condense, the nuclear envelope goes away. You start to get the structures, these spindle fibers that are forming in this prophase as well. Metaphase. Now, we call metaphase simply because the chromosomes line up in the middle, the middle of the cell. That's the key piece that happens here in metaphase. You've also got these spindles that are now attaching to that centriole, the very center part of these two chromosomes together, because they're going to pull it apart. Okay? So they're attaching to, they're getting ready for that next piece of mitosis, which is anaphase. In anaphase, those chromosomes get pulled into two pieces. And they're two identical copies, right? That's what the DNA replication did. It made us two complete copies of this DNA. Half of that DNA goes to one side of the cell, half of the DNA goes to the other side of the cell. Does that make sense? And then last but not least, the telophase is where you actually have the DNA or the chromosomes gathering at the poles, at the ends. The nuclear envelope starts reforming. The chromosomes will expand again. Your book says decondense, which makes me chuckle. It means expand, right? It gets bigger, right? So that you're back to two normal cells. So, prophase, chromosomes condense, nuclear envelope goes away. Metaphase, they line up in the middle. The chromosomes do. Anaphase, they get pulled apart. Telophase, we're reforming the nuclear envelope, the chromosomes expand again, and life gets back to normal in the cell. Question? Yes, excellent. It's like you read the next slide, Casey. It's well done. Cytokinesis is what we call that, where basically we focused on the nucleus, right? All of that mitosis stuff was focusing on just the nucleus, right? The chromosomes and the nuclear envelope. The other stuff at this point now has to get split, right? So cyto meaning cytoplasm, kinesis meaning to cut, to move. So therefore, you're dividing the cytoplasm into your two different cells. So that way you have all of the organelles, all of the ribosomes, and all the Golgi apparatus, and all that kind of stuff in the two different cells. It's a good question. Are you with me? That cell then, there's a little furrow, little hole that starts down the middle of that cell. Then you can see that there on page 129, the cleavage furrow, they call it. And then, voila, you have the breaking of two cells. Question, sir? There is the process of doing that, yes. When it's preparing for this mitotic phase, there are it will reproduce those things, but there's enough of them that you can kind of, you take half, I'll take half, and we'll make more as we're going. Yeah, that's no problem. There's enough of them there. So there's a whole big piece about cells being regulated, about how they know when they should divide or when they shouldn't divide. I'm not really too interested in you learning all those pieces. You have a question, Diamar? Okay. And I wrote on your study guide here that they're just kind of controlled by these three pieces. I'm not interested in you learning all of these different reasons of how they are. But they kind of have a timer. And there are two pieces that play a role in that. These cyclins and the CDKS, the cyclin-dependent kinases. The point of these guys is it helps the cell to know when it should reproduce and when it shouldn't. And we also have these little checkpoints. And they're in the G1 and the G2 phase. That's basically saying, hey, you're ready, you're good, you're ready to divide. Or, no, you're getting a little old. Why don't you just go over here and get into that G0 phase and you don't need to divide. Does that make sense? I'm not looking for you to understand all the pieces behind that. It's not relevant to what we are doing. But do you understand that if cells are not controlled in how they divide, that causes a problem? What do we call that when cells are dividing uncontrollably? It's cancer, right? That's all it is. It's not some bugaboo, some, I'm a terrible cancer cell. No. It's one of your cells that are just freaking out and not being controlled. It's a problem, right? What's that? Right, these regulatory pieces are not doing its job. You're exactly right. Sir? Sorry, Amar.
So I've always been kind of about cancer, right? So since your own body turning on itself, how come it's so hard to get your body to go back to healthy?
It's a good question. It's not really turning on itself. It's just that you have cells that are replicating in places and in amounts that it's not supposed to. So your body still sees it as being your tissue. It's not like it's some foreign invader that it has to fight. It still sees it as your tissue. The problem is just going too much. It's doing more than it should. It's just been damaged in these cancer cells. Yeah, whether by genetics or whether by some environmental factor. Yeah, sir. Yeah.
How is it so harmful to your body?
I guess because of where they reproduce. So let's say I have some cell in my brain that supports the brain. It's not the neurons itself. It's one of the gliocytes in my brain that helps support those guys, right? And suddenly I get a whole bunch of those that reproduce a whole. Well, now I get what's called a tumor, right? A tumor is just this big blob of cells. Well, that's pressing on the other ones and they can't get blood vessels. It's actually stealing blood from them because it grows its own blood cells because it needs more. So it's just cells that are, it's overwhelming the body. It's messing up that homeostasis.
Something like skin cancer.
Mm-hmm.
Is that like you have like more skin cells?
There are certain types of cells, whether they're melanocytes or whether they're actually squamous cells. We'll talk more about this. But yeah, different cancers have different names to them, and that just tells you what type of cell was originally infected. That can then spread other places. That's what a metastasis is. It started here, but now it's in my brain. Or started in the breast, and now it's in my lungs or in my kidneys. Make sense? Sir? Oh, the point is it would just move into that G0 phase. It would, if you get to those places and it says, nah, you're not good, it's going to make you stop reproducing. How what?
How does it make you stop?
It basically just won't let it get to that mitotic phase. Yeah, again, these pieces that control it, the checkpoints or, the checkpoints or the cyclins and the CDKS, they just won't let it go through that. And again, I'm not interested in you all understanding the whole process behind that. There's more detail in your textbook in here if you want to check it out. But the point is that it is regulated. That's the whole key I want you to understand. It is regulated. Marlon, thanks for being patient. Because the cell itself gets damaged in some way. Whether the DNA, so maybe in your genes, like let's say if you have a breast cancer, you have a genetic predisposition that those cells will produce cancerous cells. That's one way. Let's take skin cancer. You love to sit out in the sunshine and get burned in the sun, right? The UV rays actually will change the DNA of some of those skin cells to make them cancerous. And so then they'll start reproducing in ways they're not supposed to. The same. So this would just be in those actual cells of the pancreas, there's been something that's happened, whether it's genetic, whether it's environmental, from things that we ate. There's some things we know of, there's others we don't. Like we're very good at saying, oh, that's that kind of cancer. But what caused that? Oh, that's a very tough question. Because it's multifactorial. It's lots of things. It's very hard to say, oh, it was because of that one something that caused it. Some cancers we can do that. Others, we don't. And our medicine is, again, it's very basic. What do we do to cancer? We cut it out. And what do we do then? We shoot radiation or do take chemotherapy pills that will hopefully kill those cells more than they'll kill the rest of you. That's really what it's all about. Yeah, yeah. That's what you're hoping. Right. Yeah, it's almost barbaric, but it's all we got. You know what I mean? Question, Jess? And then I got you next. I'm not necessarily worried that you understand all the different phases here. I think I just simply listed them in your study guide, right? Yeah. The G phases are basically the cell living as it's normally living, right? Just doing its normal work. The S phase is where it's actually replicating its DNA. But that's still while the normal work of the cell is going on. Interphase is just the in-between time, right? Before I'm in the mitotic phase. Please, yeah, it's fine. Yeah. All right. Eloise, sorry. Thank you.
So, what's the difference between cyclins and
Come see me afterwards. We can talk more about that. I just want to keep going. Again, I'm not interested in you all being too versed. You can check it out. The textbook talks about them, but I want to move on if you don't mind, because I think we can get this last piece done about heredity, and then we'll be done as far as me yakking at you, which makes me happy. So stand up, because we got about 15 minutes. Second period, 45. Yes, good. Stretch. I've reached my limit of 15 minutes before I got to get you moving. Right? Ooh, good. Right. So let's talk a little bit about heredity, which is just basically your parents transmitting their characteristics to you, right? Through their genes. And we make what's called a karyotype, where we take all 46 of our chromosomes, we put them together. Remember, they're paired, so therefore it's 23 pairs, but 46 actual chromosomes. And we organize them by size, is really what we do, to make this lovely. And the biggest one is number 1, the smallest one is 22. Our 23rd chromosome is our sex chromosome. And not meaning the chromosome that makes you want to have sex, but the chromosome that determines what sex you are. It's a little bad joke, sorry. The 22 pairs that are not the sex chromosomes, we call those autosomes. And then the 1 is called the sex chromosome. Okay, so autosomes are all the other 23s. They all look alike. They all have genes on them. But the X and the Y chromosomes, that sex chromosome, that is a different something. Karyotype, you've got two different types. You have a diploid and a haploid. Diploid cells are what most of our cells are, right? They have 23 pairs of chromosomes. Diploid, right? 2. Excuse me. Okay, sorry about that. And the haploid cell, that means it only has 23 unpaired, right? So again, this would only be sperm and egg cells that would have that. When we talk about genes, we're talking about alleles. Alleles, different forms of genes at the same location or locus on 2 different chromosomes. So we've got dominants and we've got recessives. And you've seen this before, right? These lovely Punnett squares where we take, if a dominant is present, then that corresponding trait is usually seen, right? It will mask, even if there's a recessive allele that's present, it'll mask it. The dominant will take over, right? And what is often the case is that dominant allele is responsible for making the protein that shows that trait. Does that make sense? So the protein that gives your hair the darker color, right? The protein that gives your skin the certain color. Does that make sense? The recessive allele, again, is only seen when you've got 2 recessive genes together, 2 recessive alleles together. All right? And they often will make a non-functional variant of the protein. In other words, the protein doesn't work as well in the recessive. That's why it's recessive. That's why we don't want it, per se. Or why it's not expressed. Okay? So if you're homozygous, that means you've got 2 identical alleles. If you're heterozygous, that means you have 2 different alleles in that gene. And the phenotype is actually telling you what is the trait being expressed. So the genotype is the actual alleles, the genes, but the phenotype means how does it look actually in the person you're looking at. The trait being expressed. And so we use these Punnett squares to show possible genotypes and phenotype outcomes when we know the parent's genotype. Okay? So for example, here's a cleft chin, right? We've got cleft chin, no cleft chin. You all know who these people are? They went old school. Who's this? John Travolta, well done. And Arnold Schwarzenegger. So this cleft chin, you see that it can have either 2 big C's, right? 2 dominant alleles, or it can have a heterozygous, big C and a little c, right? And even if you have a big C, little c, because that big C is dominant, you are still going to express the cleft chin, right? A big C, little c is still going to give you a cleft chin. Even though you have 1 recessive, the dominant overpowers it. It's only if I get 2 little c's that I don't see the cleft chin. Does that make sense? Let's do a couple more of those, because again, I want to make sure you're comfortable with these alleles and these phenotypes and genotypes. So this would be CC, C little c, big C little c, and little c little c. So what's the chances of you actually having a cleft chin here? 3 out of 4, right? So 75% yes. And then the only one that's a no, right, is here. Does that make sense? Because again, the big C is dominant. All right, let's try this one here. I've got both parents. That's the same thing, isn't it? Sorry, there are supposed to be differences. All right, how about this one? So these are both heterozygous or homozygous? Homozygous. They both have the same. They both have, well, one has 2 big C's, one has 2 little c's. So what are my offspring going to look like? They're all going to be this, right? So what's the chance of them having a cleft chin? 100%. They're all going to have a cleft chin. It's just There's no one who will not. No. Yeah, unfortunately, no. There's no possible way, because they have to take one of these alleles from their parents, right? Maybe they only got this one from their dad. But there's no way that they could possibly get these two to go together. Does that make sense? And because that big C is dominant, it overwhelms it. Okay, so again, that would change what the actual genotype is. So yes, that could occur. Okay, I'll give you that. Very, very rare, but it could happen. Mm-hmm. Sure. Well, again, that can be that there was, so that we'll talk about eyes. Eyes take multiple different things to cause the color. It's not just a one gene thing. And if you have all these different convergences, sure, it could happen. Because, again, somewhere back in the line, they had to have had some DNA that was present that didn't have black hair, right? And when this baby was born, it happened to have got that gene. Or they weren't the actual parents. That would be the other one. But again, if you know they are, there had to have been something. Does that make sense?
Is left-handed, right-handed based on genes, or is that?
I don't think that they have nailed down precisely what causes that. I don't know. We have to look that one up. I'm not sure. Other questions here? I want to try and keep going quick. Last one. Shh, shh, shh. How about this? All little C's. Are we going to have any cleft chins? No, no cleft chins here, right? Any possibility of them having a cleft chin? Really, no. Okay? Good. All right. Okay, so are you comfortable that this kind of one gene to one characteristic is very simplistic? That's not actually how it works for much of the characteristics of our body. Much of our phenotype is not just one gene to one characteristic or one trait. Sometimes multiple alleles can cause the same trait. Okay? So for our blood type, that's one thing. Sometimes we need sometimes two different alleles will both give the same, right? Sometimes you've got this incomplete dominance, where I've got, again, a heterozygous, right? It's not homozygous, it's one of each, right? That they end up with kind of this intermediate trait, like in between the two pieces.
So let's say like your parents both have like the trait of sickle cell,
Mm-hmm.
but like none of them actually have it, and they have kids who have sickle cell.
Right.
But like, it's like one of them, it's like a big C, little C.
Right. So in other words, the big C, so sickle cell is an example of a recessive trait. So if you are carrying the little c, but you have one big C, we're using those letters, then you will not have the disease. But if your child got the little c from you and the little c from their mother, then they would have sickle cell, even if their parents don't. Because their parents were carriers, is what we call those. They carry that recessive gene with them. There's another one called polygenetic inheritance, that again, you need genes at 2 or more places to give you one trait. Does that make sense? It's not just a one-to-one relationship. You need multiple genes to do that. And the pleiotrophy is where one gene produces multiple effects. So you can have 2 genes giving one something, or you can have more than one gene that gives you multiple different effects. Here's some examples of that. The polygenetic inheritance, eye color is not one gene that causes it. It's multiple pieces. Okay? So this is where you say, hey, I've got 2 brown-eyed parents, and brown is a dominant, and suddenly I have a blue-eyed baby. Well, what happened? Well, somewhere back there was these genes, and they had to all line up to get that baby that had the recessive gene. Yeah. Yeah. Sure. Yeah. And eyes do change. Eyes do change. Here's a pleiotrophy. I can't pronounce this word very well. Shh. That again, one gene causes multiple phenotypes, multiple expressions of different traits. I'm cruising and I apologize. Want to make sure you see this. Some traits are carried only on the X and Y chromosomes. They are sex-linked traits. So color blindness is an example, okay? That the recessive color blindness is only on the X chromosome, right? There is no color blindness trait that will be carried on the Y. So therefore, color blindness is more common in men than in women. And you say, well, why? Because women get 2 X's, right? And color blindness, again, is a recessive. So if you have a normal non-color blindness gene on one of your X's, ladies, you can see all the color, right? But a guy only has 1 X chromosome. So if he unfortunately gets that recessive chromosome, he's stuck. Does that make sense? If he has a recessive eye color blindness, then yes, that's correct. All right, friends, we'll see you tomorrow.